Good morning, class 12th. Welcome back after a break which we have got for the test and after that a rest for 2-3 days. So let's continue with the chapter which we were discussing earlier, that is the innovations in biotechnology. And today we shall be discussing about the innovations in healthcare. Okay? Innovations in healthcare. Healthcare means in medical field, that means to take care of your health. So all these will be coming under the innovations in healthcare heading. So first topic which will be discussed here will be recombinant hepatitis B vaccine, which is produced by the gene HBsAg. Okay, hepatitis HBsAg here stands for hepatitis B surface antigen gene. Okay, HBsAg, you have already studied it, I think, in the abbreviation list. HBsAg is hepatitis B surface antigen gene. Clear? Okay, so hepatitis B, as you all know, is an infectious viral infection that attacks the liver and can cause acute and chronic diseases. The virus called Dane virus is most commonly transmitted from mother to child during birth and delivery, as well as through contact with blood and other body fluids. So it can be transmitted. This hepatitis B disease is transmitted by unsafe blood transfusions or not checking the blood for HBsAg, this antigen for this virus, for this Dane virus. If it has not been conducted, the testing thing has not been conducted, so this virus may enter in the transfused blood. Plus this virus can also be transmitted from a mother to the child. This is a very important factor here. WHO estimates that in 2015 257 million people were living with chronic hepatitis B infection. Okay, so this is alarming factor that 257 million is a big number, you see. Hepatitis B can be prevented by vaccines like other diseases, and the vaccines are safe, available and effective. Now coming on to our topic, main topic, that is how to produce the recombinant hepatitis B vaccine in the lab. Okay, so I have already told you that hepatitis B virus is the Dane virus. It has the surface antigen. Hepatitis B, the surface antigen present is isolated from the hepatitis B virus. See, you can see in the diagram, the first diagram is of hepatitis B virus. The second is for the isolation of isolated antigen gene, that is that HBs antigen gene. Then it is being inserted like other, you have studied in Superbug, the recombination technique. The same technique is being involved here, and the gene of interest is inserted into the yeast cell. Okay, now after that what happens, the modified yeast cells or the transformed yeast cells are introduced into the bacteria. And when bacteria, every time it replicates, every time it divides, it is producing, along with it, the recombinant DNA is being also replicated and being formed. So the vaccine can be taken out, collected from these and can be stored. Then this is one of the methods through which you can get pure HBsAg, that is the pure hepatitis B surface antigen. Now, as you know that in all vaccines, these antigens are injected into the body, and they produce the antiviral substances in your body. So they produce the antibodies inside your body in response. You have studied immunology in class 11th. In response to the antigens, our body produces antibodies. So once this SPHAG is injected into the body starts producing the antibodies, and hence you are getting immunization. Yes. So here it is important to note that India is the fourth country in the world to develop the vaccine of hepatitis B. after USA, France, and Belgium, and it was being produced in India in 1997 by the trade name of Guni. Gun. The word comes from the Hindi word gun, that means guni, guni vaccine. Okay, so I think this is clear. Let's move on to the next. Next is Humulin. Humulin is the human insulin. Insulin, you must have known that insulin is the hormone which is produced by the pancreas to uh, regulate the glucose in our food, glucose in our blood actually. So insulin is very much essential for keeping the blood sugar from getting too low or too high. When it is too low, it is called hypoglycemia, and when the sugar level is too high, it is called hyperglycemia. Okay, so now one basic thing, one biochemical thing about insulin is that it is a peptide. Of course, insulin is a protein peptide hormone as well as protein produced by the beta cells of islets of Langerhans, and it is responsible for regulating sugar levels, as I told you. The normal sugar metabolism, if there is any change from the normal sugar metabolism in the body, fatigue can be there, kidney disorders can be there, and heart diseases can also be one of the effects. Generally, the human insulin consists of two polypeptide chains, uh, that is total of 51 amino acids. This is something to be remembered. Human insulin consists of two polypeptide chains, total of 51 amino acids, chain A and chain B. Chain A consists of 21 amino acids and chain B consists of 30 amino acids, and both are connected with each other with disulfide bonds. So this is something to be noted here. Disulfide bonds join the uh, two chains A and B together. A chain 21 amino acid, B chain 30 amino acids. So join together total of 51 amino acids. Now let's see how the human insulin can be produced in the laboratory. Uh, Here also you will be using the recombinant DNA technology, and what is being done here from the human. See, in this whole scenario, so many people are suffering with diabetes, and most of them are on insulin, daily insulin diet, daily insulin injections. So this much amount of insulin cannot be supplied without being produced in the laboratory. So how we can produce it in the laboratory is by this technique only. What is being done? Again, the recombinant technology. See here, the bacterial DNA and the plasmid DNA is there. We are taking out the plasmid DNA and taking out the DNA from the human pancreas cell, which is responsible for the production of insulin. Then, as you can see here, there is a recombinant DNA being produced, which is introduced again in the which is then introduced into the bacterium. The bacterium is allowed to grow for certain time, and as the bacteria divides, our recombinant DNA also divides and start producing the human insulin inside the bacterial cell. Now this laboratory produced human insulin is extracted from from these cells, bacterial cells, and are stored and are ready to be marketed. So this is a very important sequential order. You should remember the technique, how human insulin is produced in the laboratory. This is the way. The basic thing here is recombinant DNA technology. In most of the innovations, we are using the recombinant DNA technology, combining two things and letting it grow in our um genetic engineer, the bacteria. Okay.